A lot of you will be wondering about what happens after your eggs are retrieved. It's an important and precise process that all takes place in a lab like this. As soon as your eggs are retrieved, our team in the lab will be monitoring your eggs for maturity and their readiness for insemination. First, your eggs are sorted based on how mature they appear. Not all of your eggs may be mature. In vitro fertilization, we won't know for sure exactly how many eggs are mature until we remove the layers of cells surrounding the egg at the time of our fertilization check. In intracytoplasmic sperm injection, or ICSI, we remove the cells surrounding the egg before sperm injection, so we know for sure how many eggs are mature on the day of egg retrieval. The maturity of the eggs is demonstrated by various physical characteristics. Immature eggs have an appearance that is very different from this mature egg here. You can see the difference in size and the clear difference in the outline of the egg. The mature eggs are then grouped together in a culture dish. The dish is placed in the incubator until sperm are added for fertilization. The sperm are usually added 2 to 4 hours after your eggs are retrieved. Male partners are usually required to produce a semen sample on the day of the egg retrieval. A semen analysis is done to check the sperm count and how well the sperm are moving. In vitro fertilization, or IVF, needs enough sperm moving forward vigorously to be able to fertilize the eggs. This is generally about 50,000 sperm for every 2 to 3 eggs. For intracytoplasmic sperm injection, or ICSI, sperm number and forward movement are not critical because one sperm is injected directly into a mature egg. For both IVF and ICSI, the semen sample is washed and processed to obtain the best and most active sperm for inseminating the eggs. On the morning after your egg retrieval, the lab embryologist assesses the fertilization of your eggs. For IVF, the layers of cells surrounding the egg are removed to look for 2 small pronuclei, the mature genetic material of the egg and the sperm, one for each. These 2 pronuclei, appearing as 2 small circles, indicate that normal fertilization has occurred. In ICSI, the cell layers were removed before injecting the egg with sperm. So on this day, we only need to look for fertilization. If there are more than 2 pronuclei, as in the following image, the egg is not normally fertilized and does not have the potential to develop into a normal embryo, and so this embryo would not be transferred. The fertilized eggs are placed in the incubator overnight to allow for growth. 2 days after the egg retrieval, the cleavage of the embryos is assessed. You see, eggs that are normally fertilized will have divided to at least the 2-cell stage by this time. At this point, they are called embryos. The embryos are given a grade based on their apparent ability to create a pregnancy. This grading is used to sort the embryos for transfer and freezing later. But for now, the embryos are returned to the incubator to continue dividing or growing for another 24 hours. Embryos are transferred 3 to 5 days after the eggs have been retrieved. The morning of the egg transfer, the embryos are checked and graded. By day 3, some of them should be at the 6 to 8 cell stage, which tells us the embryos are sufficiently developed for transfer. Before the transfer, the laboratory embryologist will briefly summarize all of your laboratory results with you and can answer any questions you may still have. The embryos that are to be transferred are drawn up into a catheter attached to a syringe. The catheter is given to a physician who inserts the catheter into the uterus and injects the embryos guided by an ultrasound. On the same day, the embryos that are suitable for freezing will be frozen if you desire. Embryos are placed in a freezing solution to protect them from the freezing and thawing processes. They are then placed into a freezing chamber, and the temperature is gradually reduced to -200 degrees Celsius. At some labs, there is the option of embryo culture if you desire, for embryos that are unsuitable for freezing or not showing appropriate growth on transfer day. For this option, we return the embryos to the incubator and their development is checked regularly for up to 4 days after the embryo transfer. During this time, the embryos must continue to divide and develop. They should keep dividing and developing into a stage called a blastocyst, which has many cells. Embryos that do not continue to divide are discarded, but the embryos that develop to the blastocyst stage are frozen and saved for your possible use in the future. A report will be made that will account for all embryos obtained from this process, so you know exactly what's happened and what you still have frozen at our facilities. And of course, as with all your treatments, we're here to answer your questions and concerns every step of the way.